Dr. McInnes, the theme of this series is breakthroughs, and these are transformative events in many, many fields. Tonight we're going to be exploring breakthroughs in genetics and the human genome, and how the genome, the human genome, is changing medicine. What I would like to do is to situate you in this unfolding story of genetics and the human genome. Perhaps we can just begin with a definition of these two terms.
The human genome and which other term?
And genetics, yes.
Oh, genetics. Genetics is the study of heredity, you know, why you're better looking than your father, or
Right.
predisposed to the same illnesses as he or your mother might be. So that's the study of inheritance. And the genome is the collection of all genes.
Okay.
And so humans have about, we're not exactly sure of the final number, surprisingly, but it's something around 22,000 genes.
When did this particular field develop, the genetics?
Like many scientific advances, genetics, it goes back hundreds of years to Mendel, and, you know, peas and whatever you studied in biology in high school and university. So he observed that, you know, you could influence the appearance of peas with just single events, or what he predicted to be single events, and he was correct. They were changes in genes, and if the gene changed, then the shape of the pea changed, for example. So that's genetics. Whereas the genome, the concept of the genome really arose from technology. That it was only until about mid-1990s that it was realized it was technically possible to obtain all the sequence is the word, but it really means obtain all the information that's contained in a human genome. And so once you had all of that, then you could, that's sort of the blueprint of life. So we had the information contained in all 23,000 of these genes. Now we don't know what they all do. We know we can make a prediction about what about half of them do, or maybe a bit more. We have a definitive knowledge of what about 7,000 of them do, and the rest we don't really have a clue yet.
Okay.
Yes.
Now, when you were started training in genetics in the 1970s, what would it be?
Did you have to say that?
Oh! Well, I wanted to establish what was the most important discovery that sort of influenced the development of genetics when you started out in the field?
I think maybe what I want to answer is what attracted me to genetics. And what attracted me was you could have a change in a single gene that had a profound impact on the biology of the organism. And so in contrast to biochemistry, which is everything's changing all the time, nothing's static, biochemistry, it's sort of the what's going on in the cell at any one moment. Okay? And so we still have a very imperfect knowledge of that. It's like trying to get an idea of the city of Montreal by taking a picture from the top of the city and looking at all the traffic moving and people walking and trying to figure out what's happening. That's what biochemistry is. Whereas in genetics, if you change one gene, you might get a profound abnormality, a child who's supremely smart or is disabled or whatever. So I liked the absolute nature of it. The fact you could really understand what that gene was doing by messing with it. And usually geneticists are looking at genes that have been disrupted. So when you lose the function of the gene, what bad stuff happens? That's the basic question that a geneticist asks. And so I guess I'm an absolutist, and I like that.
I was perhaps hoping that you would pinpoint the discovery of DNA, or at least the mapping of DNA in the 50s by Crick and Watson as a kind of key discovery in this.
Yeah, well, it certainly was. I was actually interested, though, in the without knowing nobody knew, you know, what DNA did at that point. I was interested before that. I was still interested in the fact you could mess around with one gene and get such a profound effect. So DNA has opened up the world of biology in a way that never could have happened before. And the obtaining of the information in the human genome, the blueprint of life, these 23,000 genes is going to have a transformative effect on biology and medicine and many other things over the next 50 years in ways that I couldn't begin to predict, but I can tell you about some of them.
This is an interesting point, because I gather that 10 years ago the draft of the genome was
You've been reading.
Was announced. So that suggests that this is an ongoing project. In other words, there's still a lot of work to be done. There are certain areas that we don't know very much about.
No, that's very true. So the actual genes are things that make proteins. And so we have about 23,000 recognizable genes, and therefore 23,000 proteins that are the things that carry out the functions in the cell, okay? That make us, active or asleep or awake or alert or smart or not. So the other 95% of the DNA, we have a very poor understanding of what it does. Right. And until two projects, or a project that began about 7 years ago was initiated, people used to refer to the rest of the 95% of the DNA as junk.
Yes.
It turns out to be complete nonsense.
Really?
Yeah. Well, of course, this stuff wouldn't have been conserved in biology if it was junk. And everybody felt that, but because they didn't know what it did, you called it junk. That material turns out to be, the early studies indicate, involved in regulating, telling a gene it should be active now or not active now, and keeping it quiet when it should be quiet, and so this is all regulatory stuff. So 95% of it appears to be involved in regulation of the 5% worker bees, so to speak.
That's fascinating.
Yeah, it is fascinating.
Okay. All right, I think Giovanna, you have some questions.
I'm going to ask you questions that are more on a personal, not personal about your life, but personal regarding your work and your specialty. And one is, when was the eureka, since we're talking about breakthroughs, the eureka moment that caused you to choose genetics as your specialty?
It wasn't a eureka moment. I had, what it was? I had a fantastic undergraduate genetics teacher at Dalhousie, and when I was doing my BSc, named Dixie Pelluet, and she was just a phenomenal teacher, and I just fell in love with the subject because of her. She drew my attention to it, and she presented it so compellingly, and she made me realize what I said before, that it's sort of the foundation of everything, and I found that attractive, I guess.
And what was the mystery that you found in genetics that has driven your research since then?
The ability to understand deeply what a gene does in an absolute way. I get, what's attractive is if you discover a gene and you figure out what its normal role is, and you determine what happens when you interfere with it. If it's got a mutation that causes a disease, for example, you've made a contribution to world knowledge forever. I mean, somebody else hasn't done. There's only one of those genes, and so if you figured it out, it's on. So you don't earn millions of dollars studying genetics. Some people do, but it's I guess what's attractive is the feeling that you can contribute to the body of eternal knowledge in this way. That's pretty cool. Whereas in biochemistry, there are people who do that, but it's vastly more difficult. So I don't think I'm smart enough to be a really good biochemist. But genetics is much more approachable for that reason.
Okay. Who were your most memorable mentors?
Two. Charles Scriver, of McGill, with whom I did my PhD.
And you now have the Alva chair.
Yes, that's right. So he was the first Alva chair in human genetics. He was based at the Montreal Children's Hospital, and a wonderful man. I was directed to him by my first mentor, who was Richard Goldblum, a name that's familiar in Quebec, who was the head of pediatrics at Dalhousie when I was in medical school. And I decided I wanted to go into research in genetics. And he says, then you should go train with Charles Scriver. And I said, who? And he sent me off to train with Charles Scriver, and of course that was transformative. And then,
in what way transformative?
Well, because to train, to learn science, it's like an apprenticeship, so you want to learn from the best teacher, and Charles was fabulous as an individual and as a role model and as a successful scientist. And then the second mentor who's been and still is profoundly influential is Louis Siminovich, who's the founder of molecular biology research, that's gene research in Canada, in Toronto, at the University of Toronto, and he was my first boss. He hired me at the Hospital for Sick Children, and just, I think, really took me to a new level of understanding about how to do research and what good research was. I'll give you one example. So I think you reminded me, I started there in late 1978. So in 1980, there were about 8 of us in the Department of Genetics at Sick Kids. And Lou walked in one day and says, all of you guys are going to become gene cloners. Okay, this is like a particular kind of isolating genes and understanding how they worked. And we all looked at him like he was mad. This was, we were all doing different kinds of genetics, but not that. This was a whole new area of technology that was just starting to transform. The people like him started to realize how important it was. And he forced us, with a little bit of kicking and screaming, into this new area. And at one point, some years later, I think about 30 human genes had been identified and isolated, and about 13 of them had been identified and isolated in our department. And that's because he came in and said, you guys are going to do this. So it was an astounding act of scientific leadership and insight to drag us into that. It doesn't happen very often.
And therefore, taking from the mentors you had, how do you prepare the researchers that you are mentoring?
I think in 2 ways. You try to give them great research projects to work on in your lab, and then you try to transmit to them the culture of what it is to be a successful scientist. It's like everything else in research, culture is everything. And so the reason that the scientists at Harvard are so accomplished is because of course they're really smart, but there's an equal number of really smart people in, your average university, or almost equal number in elsewhere. But it's the culture of excellence and the way they interact and what the expectations are that really count. So you try to transmit that to them. So the way to do that is, what's important in learning how to do research? Well, the most important thing, in fact, is what's the question you're trying to address. And I think the difference between a really good scientist and one who's less good is that really good scientist is addressing a really important question, like how do genes work, or what is that 95% of DNA that we don't understand, how does that work? That's important. And you have to ask that question at the right time in history. If you asked that question 20 years ago before you had the human genome in your hand, you'd be wasting your time. So it's a matter of teaching them how to approach questions, and it's not techniques.
Not techniques?
No, goodness, there are hundreds of technicians who are trained in technical school who know how to do the techniques, but you have to direct them to applying the techniques to the right questions.
And to see what they're looking for.
The question is everything, exactly.
Okay.
Yeah.
That's very important. Okay, I want to ask you a question now about your own significant achievements. What drew you to eye genetics? I believe that is...
It was very deliberate. Actually, I was so clinically I used to see patients with biochemical genetic diseases, like you might have heard of one called phenylketonuria or PKU. But anyway, single genes in which there's an abnormality in a single gene, and the patient is often picked up on a screening test after birth, and the disease is recognized, and you can often treat it. So these are diseases of biochemistry that are genetically based, that a lot of them we can treat, a lot of them we can't. So I studied those diseases initially, and
this was when you were at the Sick Kids?
Training with Charlie Scriver clinically, and also at Sick Kids. And then in my research, I identified some of the genes involved in some of these diseases. But when the ability to isolate genes came along, and we saw the Human Genome Project coming to look at all the genes and see how they interact, I realized you could address more important medical questions. Not to say these diseases aren't important. For the people who've got them, they're terrible. And collectively, there's about 6,000 of these diseases, so the burden for those families and individuals and society is huge. But I wanted to tackle more general problems, so I did what a scientist supposed to do when they're on a sabbatical, is step back and say, what's the best kind of research I can be doing? So contrary to what people think, that's what sabbaticals really are used for, not for lying on the beach in Havana. And to my wife's great distress, we went to Houston instead of to Paris. And I sat around for 6 months saying, what kind of problems and would I like to approach? And I wanted to focus on an organ, because each organ has its own set of diseases rather than some general question. And I decided to work on the eye because there are the eye is affected in more genetic diseases than any other